Dramatic music, dark crime scene visuals. Imagine this: a room, 3 a.m., a body on the floor, no witnesses, no cameras, no fingerprints. But lying on the ground, just one thing: a single strand of hair. And from that one hair, science can tell you exactly who committed this crime. This is not science fiction. This is DNA forensics, the most powerful weapon modern science has ever handed to law enforcement. Today, we are going to break down how an invisible molecule changed the entire justice system forever. Before we talk about crime scenes and investigations, we need to understand what DNA actually is. DNA stands for deoxyribonucleic acid. It lives inside the nucleus of almost every single cell in your body. And your body has approximately 37 trillion cells. Inside each of those cells, there is about 2 meters of DNA tightly coiled up. That's roughly 6 feet of DNA packed into something you cannot even see with the naked eye. DNA has a structure called a double helix, 2 strands twisted around each other like a spiral staircase. Along those strands are 4 chemical bases: A, T, G, and C: adenine, thymine, guanine, and cytosine. These 4 letters make up the entire instruction manual for building and running a human being. In total, the human genome contains approximately 3 billion base pairs, 3 billion letters, all spelling out who you are. Now here is the fascinating part. Every human being on Earth shares 99.9% of the same DNA. You and I, 99.9% identical. You and a complete stranger on the other side of the planet, 99.9% identical. But that remaining 0.1%, that tiny fraction is what makes every single person on Earth completely unique. No 2 humans, except identical twins, have the same DNA. And that 0.1% is exactly what forensic scientists use to identify criminals. 1 person, 1 code, 1 identity. So now the question is, how do investigators actually collect DNA at a crime scene? The truth is, DNA is everywhere. You leave it behind constantly without even knowing it. Bloodstains, obvious and rich in DNA. Saliva, left on a cup, a cigarette, a bite mark on food. Hair, especially if the root follicle is attached. Skin cells, left behind simply by touching a surface. Sweat, soaked into furniture or furnishing. Fingernails, if there was a struggle. Semen, critical in sexual assault cases. Even a licked envelope or a postage stamp can contain enough DNA to build a profile. When investigators arrive at a crime scene, the first thing they do is secure the area. Nobody enters without full protective gear: gloves, face masks, Tyvek suits, shoe covers. Why? Because the investigators themselves carry DNA. One contaminated sample can destroy an entire case. Evidence is collected using sterile swabs. Each sample goes into a separate sealed container, labeled, numbered, and documented. Temperature matters. DNA degrades in heat and sunlight, so samples are stored cold and transported carefully to the forensic laboratory. And that is where the real science begins. The year is 1984, Leicester University, England. A geneticist named Sir Alec Jeffreys is working in his lab. He's studying a protein called myoglobin, trying to understand how genes vary between individuals. And then, by accident, he discovers something extraordinary. Every person has unique patterns in their DNA, patterns that look different from person to person, like a barcode. He calls it a DNA fingerprint. Just like the ridges on your fingertip, no 2 people have the same one. This single discovery changed criminal justice forever. So how does DNA fingerprinting actually work? Step 1. DNA extraction. The forensic scientist takes the biological sample, blood, hair, saliva, and breaks open the cells. This releases the DNA from inside the nucleus. The DNA is then cleaned and isolated. Step 2. DNA amplification using PCR. PCR stands for polymerase chain reaction. Here's the problem with crime scene samples. There's usually very little DNA, sometimes just a few cells. That is not enough to test. So scientists use PCR to copy the DNA millions of times over. Think of PCR like a biological photocopier. You start with one tiny sample, and within hours, you have billions of identical copies. Now there's enough to work with. Step 3. STR Analysis. STR stands for short tandem repeat. These are specific sections of DNA where a short sequence of bases repeats over and over. For example, AGAT, 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 AGAT. In one person, this sequence might repeat 5 times. In another person, 13 times. In someone else, 8 times. These repeat numbers are inherited and they vary enormously between individuals. Forensic labs examine 13 to 20 different STR locations across the genome. The combination of repeat numbers at all those locations creates a DNA profile, a unique identifier. The chance of 2 unrelated people sharing the same DNA profile across 13 STR locations is approximately 1 in 1 billion, with 20 locations essentially impossible. Step 4. Gel electrophoresis. The DNA fragments are placed into a gel and an electric current is applied. Smaller fragments move faster, larger fragments move slower. This separates the fragments by size, creating a visual banding pattern. That pattern, that barcode, is the DNA fingerprint. Now the forensic scientist compares the pattern from the crime scene sample to the pattern from the suspect sample. If they match, the evidence points directly to that person. If they don't, that person is eliminated. Modern forensic science has pushed even further beyond basic DNA fingerprinting. Let's talk about some of the most powerful tools being used today. First, mitochondrial DNA analysis. Sometimes a crime scene sample is so old, so degraded, or so tiny that standard DNA extraction fails completely. In those cases, scientists turn to mitochondrial DNA. Mitochondria are structures inside cells that have their own separate DNA. Mitochondrial DNA is passed down almost unchanged from mother to child. This means you share the same mitochondrial DNA as your mother, your grandmother, your great-grandmother. It is incredibly useful for identifying remains from decades-old cases or mass disasters. Second, Y chromosome analysis. The Y chromosome is passed from father to son. In cases involving male suspects, Y chromosome analysis can trace a male lineage. It helped identify victims in mass graves. It helps solve cold cases when only partial DNA samples remain. Third, familial DNA searching. This is where things get really powerful and controversial. If a DNA profile from a crime scene doesn't match anyone in the database, scientists can search for partial matches. A partial match suggests the criminal's close relative is already in the database. Investigators then focus on that family and narrow down the suspect. This technique helped crack the Golden State Killer in 2018, after more than 40 years. Fourth, touch DNA. This is one of the newest advances in forensic DNA. Scientists can now extract DNA from surfaces that were simply touched by a person. A doorknob, a steering wheel, a weapon handle, even a handshake can leave behind enough skin cells for a DNA profile. Fifth, rapid DNA machines. Traditional DNA analysis takes days to weeks, but new portable rapid DNA machines can produce a full DNA profile in under 2 hours. Police in some countries can now run DNA analysis at the station before a suspect even leaves the building. Let's talk about real cases where DNA changed everything. Case 1. The Colin Pitchfork case, 1986. 2 young girls were murdered in Leicestershire, England. Police suspected a local man named Richard Buckland. He had even confessed. But Sir Alec Jeffreys, the same scientist who discovered DNA fingerprinting, was called in. He tested Buckland's DNA against samples from the crime scenes. No match. Buckland was innocent despite his confession. Police then asked 5,000 men in the local area to voluntarily give DNA samples. One man, Colin Pitchfork, paid someone else to give his sample. That man was caught bragging about it in a pub. Pitchfork was brought in, his DNA was tested, perfect match. He confessed, he was convicted. It was the first time DNA evidence was ever used to solve a murder, and the first time it proved someone's innocence. Case 2, the Golden State Killer in 2018. For decades, a serial killer terrorized California. At least 13 murders, over 50 rapes, dozens of home invasions. He had evaded police for over 40 years. In 2017, investigators uploaded his DNA profile to a genealogy website called GEDmatch. They found distant relatives who had uploaded their own DNA for ancestry testing. Using those partial matches, they built a family tree. They narrowed it down to one man, Joseph James D'Angelo. Police followed him, they collected his DNA from a discarded coffee cup. It matched perfectly. He was arrested at age 72. He pleaded guilty to 13 murders. 40 years of hiding ended by a coffee cup. Case 3, wrongful conviction overturned. DNA doesn't just catch criminals, it frees innocent people. The Innocence Project in the United States has used DNA evidence to exonerate over 375 wrongly convicted individuals. People who spent years, sometimes decades in prison for crimes they did not commit. Some were on death row. DNA gave them back their lives. Now, DNA evidence is incredibly powerful, but it is not perfect, and there are serious misconceptions we need to address. Misconception 1. DNA proves guilt. DNA evidence proves presence, not guilt. If your DNA is found at a crime scene, it proves you were there. It does not automatically prove you committed the crime. There may be an innocent explanation for why your DNA was present. Misconception 2. DNA databases catch everyone. Most countries only have DNA from convicted criminals in their databases. If a first-time offender commits a crime, they may not be in the system. Misconception 3. DNA evidence cannot be faked. In laboratory experiments, scientists have successfully synthesized fake DNA and planted it on surfaces. This is rare in real criminal cases, but it shows that DNA evidence needs to be evaluated carefully within the full context of an investigation. Real limitation 1, contamination. If evidence is not collected properly, DNA from investigators, paramedics, or bystanders can contaminate the sample. This has led to false leads and, in rare cases, wrongful accusations. Real limitation 2, degradation. DNA breaks down over time when exposed to heat, moisture, sunlight, and bacteria. Old evidence may yield incomplete profiles, making results inconclusive. Real limitation 3, mixture samples. When multiple people's DNA is present in one sample, it becomes much harder to interpret. Forensic analysts must carefully separate and identify each contributor, and experts sometimes disagree on mixed sample interpretations, which has caused controversy in courtrooms. So where is DNA forensics headed? The future is extraordinary. Phenotyping. Scientists are developing technology that can predict physical appearance directly from DNA. Eye color, hair color, skin tone, facial structure. In the future, police may be able to generate a face from a DNA sample with no suspect at all. Epigenetic aging. DNA can review approximately how old a person was when they left a sample. This adds another dimension to criminal profiling. Microbiome forensics. Every person carries a unique community of microorganisms on their body. Your skin bacteria, your gut bacteria. Researchers believe your microbiome may be as unique as your DNA and just as traceable. Nanopore DNA sequencing. Portable devices that can sequence an entire DNA sample in the field in minutes. No lab required. The future of forensics is faster, smaller, and more precise than ever before, and criminals have less and less room to hide. Now, let's bring this all together. DNA is the biological signature every human being carries. You cannot escape it, you cannot fake it perfectly, and you leave it behind everywhere you go. From a single strand of hair to a touched doorknob to a discarded cup, science can now trace you. Sir Alec Jeffreys once said that the day he discovered DNA fingerprinting was the most exciting moment of his scientific life. And looking at how many crimes have been solved and how many innocent people have been freed, it is hard to argue with that. DNA forensics is not just science, it's justice. If you found this video fascinating, hit the like button right now. Share this with someone who loves science or true crime, and subscribe because next week we are going deep into another mind-blowing science topic. The link to our last video is right there. I'll see you in the next one.